So what happens to pyruvate after you've gone through glycolysis? Well, it kind of depends on what the situation is. Pyruvate can go through one of three different pathways in general after glycolysis is done. And this is to harness more energy as you need it when you need it. So there's what you call the aerobic form of pathway. And this is when pyruvate basically turns into acetyl coenzyme A, NADH, and CO2. Well, that's if you're breathing in enough oxygen and you're getting enough oxygen to your cells like immediately. And that's what produces CO2, which is awesome to be able to see where CO2 comes from, right? Well, your muscles burn up that oxygen really quickly when you have bursts of energy. So we can also go through an anaerobic pathway and make the stuff that is lactate and NAD. Well, that NAD plus and lactate, some of that's converted back to pyruvate in your liver, but that lactate is what turns to lactic acid, and it makes your muscles kind of like achy and all that good stuff. So there's also what happens within yeast. So in yeast, we have an anaerobic fermentation that takes place with pyruvate, and that's what produces ethanol and CO2 as well as NAD+. So those of you who may be over 21 may be familiar with these types of compounds. Okay, so in anaerobic conditions, so there's plenty of oxygen around, it's used and unseen. Here's what happens. We have the pyruvate that we have, the NAD that's there, and we have coenzyme A. And with pyruvate dehydrogenase, we can package that pyruvate into acetyl coenzyme A and produce NADH. So that's the big overall reaction that's happening there. We got to look at that in more details in a minute. In anaerobic conditions, we're going to take that pyruvate and use NADH to make lactate and NAD+. So that's what that would look like. And that's using lactate dehydrogenase. And again, that can go back to the liver and you can remake pyruvate and do some more stuff with it like we're entering this cycle. And within fermentation, what we can see is that we can have our pyruvate come in with H plus and produce this guy, which is a aldehyde, and CO2, that then undergoes another reaction with NADH to produce alcohol through alcohol dehydrogenase. So we're going to come back to acetyl coenzyme A that we saw up here again, but let's go ahead and look a little bit more into detail about where we store energy. Okay, in order to do that, I hope we all realize that we use glucose in order to make our energy. And remember, we need things such as glycogen in order to make that happen. And so there's an entire area that we need to talk about, and that is how do we make glycogen and how do we store energy? So glycogenesis and glycogenolysis are the making and the use of glycogen for energy storage. So glycogenesis is making, glycogenolysis is taking it apart. So when we have excess glucose around and your energy reserves of glycogen are exhausted, then that's when we need to make more glycogen. And remember, if you recall from when we were talking about carbohydrates, glucose or glycogen is a polymer of glucose with alpha-1,4 and alpha-1,6 linkages, and that's usually stored in both the liver and the muscles. Why the muscles? Because that's what's making you move and that's where you use most of your energy, and the liver because that's what it's responsible for. When you have all of the glycogen that you need, then that's when glucose instead goes to make triacylglycerol or fat. So we have more than what we need at that point. We store it as fat for energy use later. But if you need glucose, great. The body is going to use that glycogen and rips it apart in order to use glycogen through glycogenolysis, okay? So we have to figure out how to both make glycogen and rip it apart in order to see how we can use that glucose as needed. It's kind of like it's our glucose reserve maker. So here's how we go through glycogenesis. And this is going to begin with the reaction of the first product from glycolysis. So if you have a buildup of this, remember that's part of how your enzyme slows down with glycolysis. That's one of the last things we talked about to sort of make sure that glycolysis is going as quickly as possible. So what we start with is glucose 6-phosphate, which is the product from step 1, right? So in another world, in making glycogen, we're going to instead use phosphoglucomutase, which is an isomerization enzyme, and the phosphate is going to move over to carbon number 1. So we have the phosphate from over here is going to come down and move over here. And so now we have glucose 1-phosphate. Now, this process is going to use a different type of molecule. UTP is going to be used and come out as UDP, therefore giving another phosphate to our glucose 1-phosphate. And what this is going to do is it's going to make a 2-phosphate type of reaction here with uridine coming off. And the enzyme that's used is pyrophosphorylase. And basically what's happening here is that we have an activation by UTP, or uridine triphosphate. And it's going to transfer UMP to the glucose. So notice that we still have the UDP coming out here, right? Okay, this allows the glucose to be added to the glycogen chain. So let me go down here just a little bit. And so here's how this guy works. We then have another enzyme called Glycogen Synthetase that adds that glucose to the glycogen chain. So we're going to end up with UDP in the end, but that glycogen synthetase allows these chains to sort of be linked together. So this oxygen, mind you, is connecting right here, and it's connecting to the 1,4 positions, right? And there's different ways to get that to the 1,6 position, but that's basically how we're doing things. And we end up with this huge long chain, and that is how we store our glucose for later use. Super duper cool. So now, if you store this for later use, that means that you have to be able to break off that chain and use our glucose here for energy production through glycolysis. There's a lot of G words here. They all kind of sound alike. So you might want to make sure you know those while we're talking about it too. Okay. So glycogenolysis takes that glycogen chain that has been stored for a little while or longer and uses it for different types of reaction. And what we do here is we actually hydrolyze the bonds between these two positions, meaning that we break apart that ether and both of them become an OH. So that's sort of what reaction number 1 is. And here we also add in a phosphate and use glycogen phosphorylase. So that ends up giving us a phosphorylated glucose 1-phosphate. Now, it's important to note that at the same time, reaction number 2 is taking care of anything that's like an alpha-1,6 glycogen chain that's debranching this guy, and it rearranges it so that we can continue the process of cleaving off the end. So you don't really see that so much here, but that is totally taking place. Okay. Finally, the third reaction, I should say, is an isomerization of the phosphate. That phosphate's going to move to phosphate or carbon number 6. So that's glucose 6-phosphate, which is exactly what we need to enter glycolysis. And so basically this guy then just goes over to the glycolysis pathway and continues through that route. Now, If you have a reaction number 4 occur, this can happen through glucose 6-phosphatase. Here we would have a dephosphorylation to make glucose, and that can sometimes happen. But hormones from the pancreas, like insulin and glycagon, can tell cells to make more or less glycogen. So you could actually even make this guy back to the thing that we started with, which is glucose. This is absolutely fascinating. I hope that you all are enjoying it, and I hope to see you on the next video. Thank you.